Wurundjeri people of the Kulin Nation and pay my respects to their elders, past and present, and the elders from the other communities who may be here today. Um, Please join us in welcoming our guest speaker, Dr. Nadia Rajab, all the way from the United Kingdom, who is an internationally recognized expert in the field of immunology and stem cell biology, with a focus on human macrophage biology. She's the scientific editor of Cell Reports Medicine at Cell Press UK and the founder and owner of Innately Science. She trained at the University of Glasgow and the University of Melbourne with a PhD in immunology and stem cell biology. Her doctoral and postdoctoral research combined computational stem cell models of human macrophage biology and has resulted in the creation of the Myeloid Atlas in the STEM Formatics organization platform. Dr. Nadia Rajab has published widely and held the leadership positions on the organizing committee of the University of Melbourne and ASSCR Stem Cell Conversations series. She has developed methods to derive human myeloid cells from pluripotent stem cells and has established co-culture models of tissue-resident macrophage biology in the areas of heart function, gut health, and infection. Throughout her scientific journey, she has been passionate in the communication of science and facilitating discussions on new advances in human immunity. She'll be presenting on the wonderful world of myeloid biology, words from a macrophage addict. Thank you so much, Dr. Nadia, for being here today with us.
Thank you for that introduction. So good morning from the English countryside. Thank you for inviting me to talk today. I thought long and hard how I wanted to present. I thought first maybe I should give you a long tedious lecture on myeloid biology and go really in depth into the literature, but I thought I'd behave and I thought I'd make it a little bit more fun. And instead of bombarding you with so much information, I just wanted to give you a bit more of maybe a lighter talk to end your day today and to start mine, and perhaps present you more questions rather than answers. And I think that's what science should be all about. And having said that, I think myeloid biology really is the perfect example where there is more questions rather than answers, and we probably know very little. And here's an image I've taken where just looking down a microscope lens, and I've taken this image using my phone. So it's very simple, and I actively encourage you to do it. But what I want to show you here is that it's a moment of stillness and a moment to look down that lens, regardless of cell type that you prefer to study, although I believe you should all be looking at macrophages, but we'll talk about that later. But when you look down that lens, I want you to just pause and think about, if you had the opportunity, what would you ask those cells? Some questions I've proposed are, firstly, where do they come from? What is their development? Is it important? Why are they so important in the body? What is it they're doing? And what is it they're trying to do to help us? And also, what happens when things go wrong? And we all make mistakes. Our cells are no different, and they can do catastrophic things in our body. And I think this is where you come in as well as the researchers here. It's the questions: how are you going to solve the problems that they do make? They're hoping that you can intervene and help them get back onto the right track, and we can talk about that a little bit later. So where do they come from? I found, this is just a generic schematic. You'll probably see this over and over again, and it's very simplified, and I love that because I like to see the bigger picture. But what I'm showing you here is just that general hematopoiesis, where you get your hematopoietic stem cell that can give rise to different arms of your immune system, myeloid compartment here on hopefully your left, and your lymphoid on the right. And we're constantly learning, and the diversity of immune cells and names coming up in nomenclature is probably getting more confusing than anything. So sometimes it is good to pause and look at the bigger picture, but also to embrace that heterogeneity that you will come to know and you will come to be challenged with, and hopefully come together as a community to find the purpose in that heterogeneity. But essentially, myeloid cells are that left side there. A lot of your innate immune cells there. They're the ones that are helping you in that first line of defense. And my favorite one, and no surprise, are the macrophages. But you will all find your own happy place, and hopefully macrophages. But there are different ways your immune system can help you. They all have their own superpowers, and I actively encourage you again to find the cell that excites you, and the population that will forever keep you curious. But using macrophages here as that example, I want to probably keep bringing up over and over again, because that's the easiest one for me to demonstrate to you that the complexity of your immune system will keep you on your toes. And with development, we're always looking at, again, the schematic. We see this development happening, but it isn't as straightforward as that. And we're beginning to starting to understand that complexity, excuse me. And I think we will continue to learn new things over and over again. So one day we know something, the next we don't, and we're always constantly challenged, and that's not a bad thing. But here is a schematic I attempted to draw up once, and using BioRender is great. But here I'm trying to show you that with macrophages, we're starting to understand using various techniques in fate mapping, transgenic mouse studies, that it isn't as straightforward as monocytes to macrophages. Your macrophages go through different waves of development, and hematopoiesis itself has multiple waves before birth and during adulthood. And here I'm showing you that your macrophages can arise in your yolk sac, and this can come in the first wave where you have your primitive macrophage progenitors, and they can seed in your brain, giving you your microglia. You can have your second wave that will come to your erythromyeloid progenitors, and later down the line you'll have your hematopoiesis shifting to your bone marrow, where you'll have that throughout adulthood, where you have monocytes arising and can seed into your tissues. And this is a constant argument about your tissue-resident macrophages seeded before birth. They're in your tissues. So what's the role between those and the contribution of monocytes to that population? And you can take part in that debate. I don't think there is a right answer there. What is important about those cells that are seeded before birth? And what is the contribution of those that are contributing to those populations? I love that debate, and I hope you'll be involved in it as well. But I think that brings me to one of the first questions, or set of questions I want you to start thinking about. And again, I'm not going to give you answers because there are no answers to this, but questions I actively think about quite constantly. And firstly, does development matter? We see different waves of it occurring. We see, again, macrophage arising from a non-monocytic progenitor, and those that do arise from a monocyte progenitor. Does it matter? Does that development define a particular cell's identity? And regardless of cell type, this can impact any cell type you're particularly interested in. Does their development impact how they function? What is the role of development here? And this is important when we start thinking about ways we can model these cells, and we'll again come back to this very soon. And something everyone can debate about, but I just want you to start thinking about that. So why are your myeloid cells so important? I've used your macrophages here as an example because they're a perfect example of their importance in inflammation, their production of inflammatory cytokines, the phagocytosis, the cleaning up of debris, and their orchestration of wound healing, their production of anti-inflammatory factors, and they're doing all these activities based around various tissues in your body. And this is not only the important aspect of your myeloid cell compartment. Your myeloid cells are that first line of defense. I think I already alluded to the involvement army tactics here in the immune system. Your first line of defense is so very important. They're the ones kicking things into action, alerting people to the rest of your team, your immune system, that something is attacking. We need to kick into gear and let them know that there's danger. And that comes down to the fighting of infection. They're the ones that are pretty much taking the brunt of this. They're fighting away while your adaptive side is kicking into gear. They'll process that antigen and they'll present that danger, that message, and that signal. They're initiating that inflammation, not maintaining that dangerous environment. And they're the ones that will orchestrate that resolution as well. And that's so very important. And in turn, at the end, that wound healing, the production of growth factors that will help maintain and encourage that to happen. And I think, I don't know if I cut off, but it's a fun fact that it's in macrophages that, for example, when you have a tattoo, that is a maintained wound essentially, and they're the ones on the reason why your tattoos will persist. And my personal favorite is the capacity to remember. And this is something that's so fascinating to me, because the adaptive immune cells, and even when I was studying immunology back in the day, it was, adaptive immunity is the ones that have the memory. And the innate immune cells were still just the ones that are saying, oh, we see some enhanced responsiveness, but that memory aspect was not really appreciated. And I think now, thankfully, we're starting to really appreciate that your innate cells, and particularly in this case macrophages, because I love to study them, have this capacity to remember. Right, so they will see something, they'll remember what they've seen, and they'll change how they respond the next time they see it. And we're starting to understand that memory is actually developed throughout their development, and it can actually occur even at a progenitor stage, and even potentially passed down to another generation. So it's an educational process development, and maybe that's something to think about as well. Sorry, it's a bit slow on my side. But, we all make mistakes. Myeloid cells are no different. Unfortunately, they do naughty things. And we have seen so much in the literature now, just a Google search will tell you that myeloid cells are implicated in a lot of awful things. And they are perhaps, orchestrating that chronic inflammation. They are in overdrive production of cytokines. We have sepsis, where that could be that over response, but it could also be that memory aspect where they're no longer capable of responding to infection. So that's really interesting as well. And we do see a lot of neurodegeneration coming up in the promotion of cancer, diabetes. The list really does go on. And it's so easy to question whether myeloid cells are they a blessing or are they actually a curse? I'd like to think they're a blessing, but many would argue that they are perhaps borderline on a curse as well. But they are there and they mean well, but they can do bad things. And I think this is important because these can tell you where you can come in, and this is something to reflect on as well. And I think this is important regardless of the cell type you look at, whether it is the myeloid cells or it isn't. And the questions you should be asking, I hope you're asking, is how can I study these cells? What models do we have to study them, and can we what can we do with that? Is it are we going to try and target the cells? Are we going to look at them to target surface receptors, or are we going to go intracellularly? Are we going to start looking at their signaling components, or how about the metabolism? Can we rewire them? Can we go transcriptionally? Can we gene edit? Can we epigenetically target them? The list can go on, and there's so many beautiful techniques coming out now where you can try different things. I propose a designer cell where you can perhaps design a cell that can potentially function the way you want them to function. And I don't think that's going to be too far off. And we're starting to see some literature describing the development of cells that with a particular function, or potentially to deliver certain goods to different tissues. And it's a really exciting time to be part of this. But with all this, you're essentially asking, how can I develop ways mistakes they have made? And with the mistakes, we can learn a lot about them. I don't think it's all a bad thing, because despite the fact that we want to reverse the mistakes that they make, we can learn a lot about these cells through the mistakes that they do make. We shouldn't be scared of the damage. We try and find the good within that, and ways we can perhaps train them to learn and to maintain the education that they should have become accustomed to. And with that, we need to ask about how we're currently studying them, how we're learning about them. And a lot of immunology, it should be no surprise to everyone here, is that most of what we understand from immunology, immune cells, has come from animal models. And we're starting to understand that there are so many differences between man and mouse, or regardless of animal model. Fundamentally, we're very different in regards to our genetics, and that can transpire to a lot of different components. So they're not exactly the best model to study this. Regardless, they have provided us a lot of information. So I'm not going to knock that on the head. But they're also immortalized cells. Again, in the literature, we see a lot of this. It's a really, I guess, an easy thing to use in the lab. They're very hard to kill. So in cell culture, it's great. But unfortunately, they're not physiologically relevant. Again, not the best model, regardless of their capacity to look at responses, etc. But even looking at it from a macrophage perspective, these are probably monocytic lines. They're not mimicking that tissue residency. Again, the development is not the same, and they're essentially cancerous. Right? They are overgrowing, constantly growing. They're not mimicking the biology that perhaps you'd want to mimic. And peripheral blood has again been a really useful source, and I actively use peripheral blood. I thought it was fantastic, mainly to get monocytes. Again, if you're interested in harnessing monocytes or different components of your blood, they are great, and they are from a human source, right? So that's the closest we can get without damaging transcriptionally, because if we try and get tissue resident cells, even the extraction process can really change how they transcriptionally look. So trying to develop a way to model cells is a very big challenge. But again, peripheral blood has its own limitations. Your immune cells that you get have a limited lifespan. They have a limited source potential where you're gaining it perhaps from a donor you won't be able to go back to. And there are, again, developmental limitations when we start thinking about whether we want to mimic that, the various developmental routes that we can get ourselves. Macrophages, do you want to mimic that development where you get the macrophages from a monocytic progenitor? Or are you more interested in a more primitive development instead? But what's been really interesting, I think it's going to cause a lot of change, is that the FDA did announce that there will be less requirement for animal tests before human drug trials. So with that, there's been and will be an increase in using different models, and more particularly the use of pluripotent stem cells. And the use of pluripotent stem cell derived models have a lot of different advantages. They are going to be your human relevant models because you are obtaining cells from a human, and we can make that, whether it's skin biopsy or even blood, we can rewire them, reprogram them back into a pluripotent stem cell state. And with that, we can give rise to any cell type we want. Again, we have this capacity to have a continuous source of cells. They are stem cells. They're going to grow and grow and grow. And we can maintain that same genetic background, which is great. So that source issue is overcome. We have the capacity to genome edit. So we can start doing knockouts. We can edit, target those kind of aspects. But also genetically, we have an opportunity here to study immune cells that from people that are perhaps suffering from genetic disorders as well. So if they're impacted their immune system, we can model that. And again, we can look at development. Development is always going to be a question that's going to continuously come up. And I'm going to sound like a broken record, development. But for me, it's a very important thing to think about because it's something that people are arguing about. And with stem cells, you are essentially giving them proteins or instructions that will dictate how they develop. So whatever instructions you give them, they will respond to. So with that, we have a capacity here to look at that stages of development and see how that actually impacts the cells that you make. So macrophages, again, of course, they are the fantastic example, and I'm going to keep bringing them up and up and up. Don't worry about all these on the right. I just thought I'd give you some fun literature to look at. But these are a lot of the protocols that are up. This is not limited to this. They're continuously coming up. A lot of them are based on perhaps the Van Milgenburg paper. But overall, and essentially what they're doing is giving your stem cells, whether they're maintained on those feeder layers, we're trying to go feeder free, and they can be formed into embryo body structures, so these 3D structures, or they can actually be differentiated flat in a 2D like phase. Regardless, you're differentiating and providing the factors that will dictate that differentiation and giving rise to epigenesis in your culture that will, what you propose are going down a developmental trajectory down into a monocyte-like cell, or if not, going towards a macrophage stage, regardless at the end. This is super fun. I really encourage you to give it a go and fun fact, when I first tried to do it, I actually thought it was not going to work. I forgot my dish in the incubator. And after, I don't know, I think it was a couple of weeks actually, I had to look. It was very naughty of me, but I actually did look under the microscope before I threw it out, and actually had macrophages. So my take-home message there is, don't throw your dish without looking at it. And the best things happen by surprise, I think. So don't doubt yourself, give it a go, and you're more likely to succeed than not. So what I used to do is have my stem cells form that embryo body formation. My method was using more spontaneous aggregation because it was just so much easier rather than going down the different routes that you can do embryo body formation. But what you get here in the stage 3 is what I consider a galaxy. I thought the planets and these beautiful stars around them. Yep. So essentially giving them instructions that go down the different days of development. You're getting a mesoderm induction, you're giving rise to hemogenic endothelium, and essentially there's progenitors that I'll pump them with MCSF, a classic macrophage differentiation growth factor. And essentially you're going to get these beautifully slug-like cells. And these are fantastic to look down the microscope and very deceiving because they look very still, but they're actually not. But one of the questions, again, I'm posing to you is, what makes a macrophage a macrophage? And again, that question could go to whatever cell type you're making. What is it that makes that cell type that cell type? My thing I say is, it looks like a duck and it quacks like a duck, but is it actually a duck? So, you when you go in the lab, you have your cells, you're more likely to not look at the morphology and you think, yeah, all right, they look like what I want. You're more likely to go down the flow cytometry route, and they look at surface markers. We all love that. And here what I'm showing is a few different cells in comparison. I used peripheral blood monocyte-derived macrophages as my benchmark cell type, and I had various stem cell-derived macrophages that developed in the lab, and I compared them to that peripheral blood monocyte mac. And generally, the population seemed to have very similar expression of surface markers. You're looking at your CD14, your 16, your 11B, CD45, no surprises there. But yeah, MHC, I threw that in because I was curious. And I'm glad I'm curious because, that shows us there are differences there. And this aspect is that there seems to be perhaps a naivety in the cells that you're making. They have very low expression of that MHC. There might be a maturation thing happening here that needs to be done. But, you could think about the environment and the way you make cells and how myeloid cells are very sensitive to the environment. And they do respond to the cues that they received in that environment. When we think of peripheral blood monocytes, we're taking that from an in vivo environment. We don't know what they've seen. They've seen probably circulating endotoxin. They've gone through that development in vivo. They've seen a lot of things. They've already had a pre-education in my eyes. So we have a very sterile model system here using stem cell macrophages in a dish. They only see what you give them. And I think that could be something used to your advantage. And you can look at ways we can shape these cells in a controlled environment as well. I've added this video here because this is one of my favorite ones, because these are stem cell macrophages where I was looking at how they can collect up these pH rotobeads and where they would phagocytose them and put them into their acidic compartments, turning them red. So it shows that they have the capacity to take up these beads and attempt to break them down. And I love that because when we have these little cameras in the incubator, or looking at live imaging, it really brings your cells to life. Looking down the microscope, they are very still and it's very easy to dismiss them, but they're doing so much. And having that live imaging is so exciting, and really actively encourage you to do that too. So what makes a macrophage a macrophage? And again, more questions for you. These are important ones because, again, there are no right answers, and it's so easy to get so overwhelmed with ways we can convince other people that we have the cell types that we have. But the question is, what makes a cell a particular cell type? What is it? Is there a right answer to this? Is it their surface markers? Is it what they do? If they're able to model something that you want to study, is that sufficient? What is it that's going to convince you that you have what you think you have? I think that's probably the most important one, because it's, again, very easy to go down a hole and to doubt yourself. But again, have courage. You probably have what you think, but if you don't, I think that's even more exciting. But also when you're going towards that publication route, you're going to have to try and convince other people. And that's when it gets a bit more challenging. And we all have our favorite benchmarking techniques. We all have our own benchmarking requirements. But if that's how you want to benchmark and you propose that, I don't think there's an issue with that, because you are saying, this is the requirements I need in this cell type, and this is how I want to benchmark them. And this is how I think I've got the appropriate model to study what I want to study. And I don't think there's a problem with that. But again, that's something that people are still debating, and we always have a new CEPF marker coming up. We always have a new transcription factor to express, and the list will go on and on and on. But don't be afraid, and just try and think about what you think your benchmarking requirements should be. So now I've come to maybe a little bit different, and something I want to, I think, push forward a bit more. We've always been constantly afraid of computational biology, and I know I was, and it's so easy to see it as hardcore code that I don't know what to do with. And thankfully, I think we're starting to come together. The wet lab and the dry lab are now starting to hold hands and realize we can actually benefit each other. Computational biology, proposes a hypothesis, gives us information we cannot see down the microscope, and it's super exciting to be able to look at that data and to make sense of that data. So give coding a go. I only gave it a go because someone said that I probably wouldn't be able to do it, and I'm stubborn, so I just gave it a go, and it's actually really super fun. And to have that control of your data is exciting, and it should be exciting. So do give it a go. There's so many fantastic people in WEHI itself that are fantastic computational biologists. So give it a go. See what you can come up with, because it's going to become extremely important to be able to do so. And we're starting to see a lot more computational analysis coming up. There's a lot of interesting sequencing technologies coming up and coming out. And the amount of publications now describing various transcriptional techniques. We're going single cell. We're looking at that resolution. Bulk-seq is forever to stay, I think. But there's also aspects of spatial transcriptomics, etc., that are becoming extremely fashionable and popular. But here, I just wanted to show you an example of how we can look at data and bring data together. And this was in the construction of the Myeloid Atlas. This is something close to my heart, and I actively encourage you to explore it. And this is an example of bringing public and proprietary transcriptomic data together. And there's around 900 samples, but this is constantly being updated. So look at it. If you have any data to include in it, please do so. But what's exciting about this atlas, and what was exciting for me, was that I had an opportunity here to look at the cell identity, different cell populations, how they're all perhaps transcriptionally different, and to explore why that is. What you see here, I'm not sure if I can, let's try pointer. So hope you see my pointer, but you see the spread at the top of your macrophage section, and they're coming all the way from your monocytes. These are actually your stem cell derived macrophages, and your tissue ones tend to sit around here, or your fetal microglia around here. Sometimes when they're cultured, macrophages, particularly microglia in these sample collection, they tend to sit in the spread as well. But what's interesting here is that despite the benchmarking, that seems to show similarities. And when you do your short, small scale analysis, where you just look at a couple of samples benchmarked with a cell type, you don't see that spread and that diversity as you would in atlases. And this really demonstrated that our models aren't where we think they are. They're actually very, very different in my eyes. But I think that's really interesting. I'm not sure what's encouraging them to spread in that way. There are different differentiation strategies. Assume maybe they have different impacts. But what we're seeing is that we can create a cell type, but they're not mimicking each other. And I think that's super interesting and something to consider when we start deriving these cells, and understand that they are very sensitive. And what can we do here, and what can we learn from this? Is there an aspect of the development we're not capturing accurately? Does development matter? if we're able to study what we want to functionally want to understand, doesn't matter. And these are something that's constantly on my mind, and I don't think there's an answer to it. But personally, when I've attempted to study stem cell macrophages, we do see slight differences in the ability to respond to an infection, and why that is, I'm not sure. But again, that shows the heterogeneity in the capacity of a cell to respond. I think that's something to appreciate. Macrophages aren't simple. No cell is simple. And again, that will probably come down to the educational upbringing that they did receive. So we can start asking questions. Myeloid Atlas is just one example. We see so many being published now, a lot of data coming together. And I think that's a really fantastic thing, because we can start asking those questions of how well our models are doing. We can see the differences in the cells. We can start asking what defines a cell type, what is cell identity, what influences a cell's transcriptional profile, what makes it sit in one place rather than the other, and can we learn how to design a cell based on that? And we can ask that question of whether cells actually have an identity crisis. Is there such thing as identity? And should we try and fit them into that little box? And instead, should we actually be embracing that heterogeneity and figure out what we can learn from that? So what I'm proposing really is upfront is not to be afraid of data. What can we learn by bringing data together? There's so much data out there. There's constantly publications describing the sequencing of new samples, which is fantastic. And it's going to become more and more data out there for you to repurpose and to look at and to have fun with. It shouldn't be limited to the scope of which it was probably initially sequenced for. But there are so many questions that are readily available to ask. And not everyone can ask them or have the time to ask them. And you need to find a question that's going to excite you. So how can we repurpose that data to look at myeloid cells, understand them, model them, find ways to design them? And what can you learn from the range of sequencing technologies out there when we start bringing all those various beautiful techniques together? What can we learn? And single cell technology is obviously very fashionable right now. What can you learn with that newfound resolution? And understand that purpose of heterogeneity as well. So I guess to wrap up, really, as I go on and on, and I hope I've inspired rather than confused you. But with science, I hope you'll always continue to ask questions. And I think the excitement is to ask questions and not to know the answers. So I truly hope you never do find the answers, because in that case, you'll never stop asking questions. And with that, I will stop there. Please feel free to message me at any time if you do want to talk the hardcore literature. Always happy to talk cells. Otherwise, I'm happy to try and answer any questions you have now. But thank you.
Thank you very much, Dr. Nadia Rajab, for such an interesting and very enlightening talk on macrophages. It's such a complicated cell to deal with and very rare. So if there are any questions from the audience, or even online. Any questions?
Yes. I've overwhelmed them and stunned. That's fine.
Yes, you've got it.
I have a question about the macrophage you induced in vitro. So how can you mimic the tissue environment in vitro? Because the macrophage, the tissue-resident macrophages, they really need the environmental signals to induce their functions and to help maintain the tissue homeostasis. How do you tackle this kind of problem?
It's a fantastic question. So yes, the environment's so important. The tissue environment will provide those cues. And what we're starting to trial is looking at incorporating them into co-culture systems where you have them embedded in tissues, such as cardiac tissue, etc., where we can actually see them change and adapt that tissue environment. There's also development of organoids. We're having that the more complex environments where we can culture these cells in because with a 2D structure in a flat in vitro environment, you don't have the complexity that they should perhaps be seeing. So we're starting to try and incorporate a bit more of the 3D structure types, the organoids, and hopefully that will provide some of the cues that they're missing in that 2D flat culture, but they're not perfect yet. So the more we learn about even that in vivo environment, which we're still learning about, and trying to understand the cues we need to give the cells, and that's going to be part of that education that you need to give those cells. So we're constantly learning. So my answer there would probably be co-culture and organoids and understanding additional factors we need to add into that environment.
Thanks, Nadia. We have some more questions from the audience.
Hi, regarding cell identity and the transcriptomics aspect, I wanted to know, are there any clinical applications that can arise from understanding macrophage transcriptomics?
Yes, I think so, because if we can understand them transcriptionally and what's going on, even in a disease setting, maybe that can give us perhaps some instructions and ways forward where we can start thinking about how we can tweak that. There's something that's happened that changes their transcriptional identity, and we can understand the signals that may give rise to that and ways we could perhaps prevent or reverse that. So I think transcriptionally, and that transcriptomic aspect is going to be very important to understand ways to target them.
I had a quick question about, I think earlier you mentioned sort of briefly about memory, and that during the different stages of myeloid progression, they can get memory throughout each stage. Do you think that memory would look different or have different strength or something like that, depending on what stage it was in?
That's super interesting. I love that question. Mainly because we don't really quite know, because I think there are some evidence now that suggests that memory obviously can arise early on and can be passed down towards its final cell type. And what it actually sees to begin with might actually influence how they respond downstream, whether that's perhaps more enhanced or uncontrollable. And I think using a model like iPSC macrophages in that sterile environment, we can start asking those questions, because we can start giving those early progenitors something and seeing how they respond downstream after that initial educational provision early on. So with the in vivo environment, it's very hard to determine what's happening and how that can impact downstream, because there's lots of uncontrolled variables there. But with your stem cells, you have that controlled environment, and we can start asking that question. We do see enhanced responsiveness, after vaccinations, etc. And we're starting to understand that, the innate compartment changes how they respond based on vaccination, and that memory compartment is still there. So we do see the ways that they can enhance their response already. But based on strength, etc., I think that's still an open question, and I think new models will help us understand that.
Are there any more questions from the floor right now? There's a question online from Dylan Sheeran. Great talk. What have single-cell sequencing methods taught us about macrophage heterogeneity?
Oh, I love it. So single-cell resolution has been really insightful to tell us that we know pretty much nothing about macrophages, because there's so many different little groups amongst the populations we thought were just one big bulk. Having done some single-cell sequencing in the lab myself and looked at even just our iPS macrophages, which we'd hope they've all seen the same queue, they'd be the same. But actually, we start seeing heterogeneity even within that. And what the roles are within that, we're still not sure. And there might be, different parts of cells that are doing different things in a response. We're not sure about that either. I did see aspects of proliferation happening, and whether that's something in our stem cell max and maybe mimicking a self-renewal capacity, I'm not sure. So again, more questions rather than answers, but what it's taught us is that it's more heterogeneous than we thought, and there's going to be a huge era of embracing that diversity.
Can I ask just a quick question? It's about macrophages in vivo setting and in the peripheral blood. Do you see, how do you identify macrophages in the peripheral blood? Because you identify monocytes, but in terms of actually macrophages, I know that the culture that you showed, the cell culture, which was very spindle shape, that's not actually present in the peripheral blood. But you do see macrophages in the bone marrow, especially in a macrophage type activating syndrome. So
yes, see identity. That's the question. What's going to convince you? A lot of people tend to look at integrin expression. You know, are they more likely to be stickier? Monocytes tend to lift, but you're right. You know, they can be different depending on where they're found, and how we're going to convince ourselves that we have the cell at that macrophage. That's something you're going to have to try and convince yourself with. There's going to be, everyone has their bias. I think for me, it's always been looking at them and a gut feeling, and I don't think that's very scientific of me, but it is a gut feeling. But yes, looking at surface markers can be helpful there, and again, it'll show you that diversity. And again, I think the atlases and transcriptionally benchmarking them on a bigger picture, I think that's going to help us understand that identity as well. But I know, you know, isolating in vivo cells, that's a whole challenge in itself, and to try and get enough numbers, that's another challenge too. And that's why I think people are moving towards the stem cell models instead. But I don't think that really answers your question, and I'm sorry. But it's a big challenge. But
I guess that's where the single cell technique comes in and plays a big role there, I think.
Definitely.
Yeah. It's very interesting. Any other questions online or from the floor? No? No? Okay. So thank you so much for your excellent talk, and it was very inspiring to hear you all the way from the UK. And is there anything you'd like to ask us? Otherwise, we would close the meeting today.
No, that's fine. So please do reach out to me at any time if you want to ask any questions or etc. Always happy to
wonderful.
chat. Thank you.
Okay, wonderful. Thank you so much. Oh, good. Thank you.
Bye.